Our next speaker is Dr. Shinwon Park, a postdoctoral fellow at the Autism Center of the Child Mind Institute. Please join me in welcoming her to the podium.
Hello, I'm Shinwon Park, and thank you so much for coming to this talk. Today I'll be talking about the role of thalamocortical connectivity in shaping large-scale functional brain organization. So the first part of my talk will mainly focus on typical development, presenting some of the findings from our recent paper. Understanding typical development is crucial as it provides a framework for studying atypical populations. For the second part of the talk, I will present some preliminary results that I am currently working on with autism. So, you are here in Seoul for the OHBM conference. While you could be outside touring palaces and shopping, you chose to be here listening to my talk. Now, let's zoom in and see what you might be doing right now. You are probably looking at the slides and listening to what I'm saying. Simultaneously, you may be relating what I am saying to what you already know, or you may be trying to understand my intentions. And some of you might even be daydreaming, which I hope doesn't last for too long. So my point here is that all of these ongoing processes can be divided into largely two different information processing streams in our brain, one for external stimuli and the other for internal stimuli. Based on numerous studies about the role of various processes going on in our brain, this differentiation can be captured in the cortical hierarchy of the principal functional gradient, which spans from the unimodal sensory motor to the transmodal association brain regions. And this balance of external and internal is one of the fundamental brain organizational principles. In fact, previous studies have shown that this external-internal axis becomes more segregated across typical development. As you can see here, the sensory motor and visual networks on one end, as well as the default mode network on the other, is expanding across age groups. But now you may wonder, how does this axis emerge? The thalamus is a highly viable candidate for investigation. Traditionally known for relaying sensory information to the cortex, it is now increasingly recognized for its role in processing internal stimuli. Also, research has shown that thalamus forms connectivity with different regions of the cortex at different developmental periods. For example, thalamosensory and thalamo-salience networks form early while the thalamo-default mode network components develop later. Additionally, a recent study showed that the developmental period of the thalamus is particularly protracted compared to other subcortical regions, extending up to even 7 years. And thus, we decided to investigate the development of thalamocortical connectivity and how it may influence the functional specialization of the cortex during typical development. We used large-scale open data sets that allowed us to examine various developmental stages from infancy to young adulthood, and also multifaceted approaches, including dimensional reduction to resting state functional connectivity, transcriptomic analysis, and computational modeling. But today's presentation will particularly focus on the results of neuroimaging and computational modeling from childhood to young adulthood, because this is when the external to internal axis emerges and develops. These are the first and second thalamocortical gradients and the neocortical projection maps, or neomaps, of the thalamic gradients. To briefly explain the methods, for every voxel within the thalamus, we extract its correlation with the neocortex, and then we compute a similarity matrix, which indicates how similar the functional connectivity of each thalamic voxel is. Next, a dimensional reduction process is applied to this similarity matrix, which results in a set of thalamic gradients. Then these gradients are projected onto the neocortex, so we can interpret the thalamic gradient patterns based on their neomap patterns. In both the maps, the salience network is consistently positioned at the higher end of the gradient. However, if you see in the first neomap, the visual and somatosensory networks are situated at the lowest end of the gradient, and in the second neomap, the default mode network took that position. So collectively, this may suggest that the thalamus plays a role in differentiating between the internally and externally processing brain regions. To test this observation, we performed a data-driven network profiling of the neocortical projection maps. Additionally, using a reference coordinate system where the two neomaps each serve as the x and y axis, we found that the salience network is commonly placed at a joint position that links the two other network chains of external and internal brain systems. When we examine the change in the distribution pattern of the for the first and second neocortical projection maps, we found a decreasing overlap between the external and the internal networks across age groups. In other words, more functional differentiation was found across development. Additionally, using longitudinal data, we saw a significant increase in the segregation between the internal and externally oriented functional networks across development. So now, to better understand the mechanism underlying these findings, we explored a series of generative network models. These models create synthetic networks according to predefined wiring rules. Then we evaluate the model performance by examining how similar these properties are to the actual empirical functional data. So if a model gives us a synthetic network that is similar to the empirical data, we think that the wiring rules used in that model may be telling us something about how the cortical networks are formed. Then how does the model generate the simulated networks? As you know, our brain consists of nodes and edges. Now, what the model is doing is at each iteration, it calculates which two nodes will become connected. And this wiring probability is calculated by trading off the cost of a connection forming based on distance against the value of the connection being formed. For example, I brought a cartoon for an easier understanding. For example, let's pretend that you are node 2, and in the absence of any other wiring rules, spatial location would drive the wiring, and you would form connections with the closest node, which is 36. However, if you enter in some kind of wiring rule, for example, let's say the topological constraint of matching index, then that would allow two nodes that share a lot of connections with other nodes to make a connection. So despite the longer distance or the cost, an edge will be able to form between the two nodes. And so for this study, we used 3 different wiring rules based on thalamocortical connectivity, gene expression, and spatial distance. Looking at the results, our simulations showed that the thalamocortical connectivity-based wiring rules provided the best fit, supporting our results that the thalamocortical connectivity plays a crucial role in cortical network formation. And I brought this result. We did several other, we measured like network modality and segregation indices, but I thought this was particularly interesting because compared to the other wiring rules based on gene expression or spatial distance, we derived gradients from the simulated data, and you can see that it is highly similar with the actual gradients observed from empirical data when we use the thalamocortical connectivity rules. And next, we perturbed the thalamo-salience connectivity of the wiring rules in a series of growth models based on our previous neuroimaging results. And using growth models, it's an approach informing the network simulation with age-dependent developmental patterns. And perturbations were applied to each different developmental period, 8 to 12 years, 12 to 18, and 18 to 22, or to all of them. And then we compared it to a non-perturbed growth model. So if you look at the results, we found that perturbing the thalamus-salience network compromised the segregation of both external and internal axes. However, you can see here the salience internal segregation was more dramatically affected, suggesting a higher contribution of thalamus-salience connectivity to the development of internal processing regions, such as the default mode network. And using the simulated data, we extracted cortical gradients, and you can see that there was a more detrimental impact on hierarchical network formation when applying the perturbation at later stages of development, which is consistent with the notion that the default mode network has a more protracted timeline of development. Finally, we performed several sensitivity control analysis, such as perturbing the thalamo-sensory connectivity or thalamo-random nodes across the brain, and found that it did not affect the functional segregation of the internal systems as much as perturbing the thalamo-salience connectivity. In sum, we found that the thalamus actively shapes the emergence of an internal-external cortical hierarchy, and that the connectivity between thalamus and particularly the salience network affects the segregation of the external-internal cortical hierarchy. And thus, this framework may offer insights into the pathophysiology of neurodevelopmental disorders shown to have atypicalities in the externally and internally oriented information processing systems. So our next step is to apply and extend this framework to better understand autism. In fact, one of the core features of autism is the simultaneous presence of atypicalities in both sensory motor processing and the higher order socio-cognitive functions, and on top of it being such a heterogeneous clinical condition in terms of intelligence, language, comorbidity, and so on. And thus, the thalamocortical gradients may provide a unified framework that allows us to link these different phenotypical patterns to a broader system-level imbalance. We could particularly find like a disruption in the macro-scale hierarchy that affects the integration and segregation of these external and internal functional systems. Yeah, so a previous study found less segregated cortical functional gradients in autism compared to neurotypicals. Particularly, you can see in the figure that the internal-external axis is compressed in autism. And now that we have an understanding that thalamocortical connectivity underlies the emergence of such cortical functional organization, and it has not yet been examined in autism, we thought, yeah, we should do it. So we used the ABIDE data set to extract thalamocortical gradients, and we found significant group differences in the first gradient map using FDR correction. And looking at the neocortical projection maps, you can clearly see a gradient suppression in the autism compared to the neurotypicals. And significant group differences were found particularly in the insula and superior temporal regions where autism showed lower values. Moreover, we found a significant association between the ADOS total scores in the values of the insular region, mainly comprising of the salience and sensory motor functional networks. So, in typical development, the thalamo-salience connectivity was an anchor for differentiating between the external and internally oriented functional systems. And although further analysis is required, group differences and these significant clinical associations within the thalamo-insular region suggests that atypical connectivity may underline the compressed external to internal axis in autism. However, these results are based on a wide age range, and thus, in light of the dramatic developmental changes that occur before school age and the early onset of autism, there is an urgent need to study more early developmental stages. And sleep resting state fMRI serves as a window to elucidate brain changes in early developmental stages, as it helps to mitigate the motion-related artifacts commonly found during scanning of young children. But we should first try to see if we can compare the different states of sleep and awake, because a lot of the scans conducted in older populations are during wake. So using a within-subject data set consisting of 2 MRI sessions, one in wake and one in natural sleep within the individual, we examined for any potential state-dependent effects. And high spatial correlation was found between the thalamic gradients obtained from awake and sleep at both the group and individual level. So we can see that there is no complete reorganization of the essential network configuration. And finally, we started to investigate the thalamo-gradients in the context of long-term longitudinal design of autistic children. And as the data collection for this project is ongoing, these are the preliminary results of the thalamocortical gradients extracted from the children with baseline MRI data. And comparing it with the overall patterns of the thalamocortical gradients in the typical development, you can see that it's very similar. But however, since the age range is different from our previous study, we will need to obtain more age-matched neurotypical controls. And at the moment, we are collecting the longitudinal data, and one of our goals is to examine the brain behavior relationships within the autism sample to better understand potential cascading effects of atypical development in low-level sensory networks and how it may influence the development of higher-order social functions. And so I guess my 3 take-home messages are that thalamocortical connectivity plays a crucial role in shaping functional cortical organization across development, and that understanding of typical development provides a framework for studying atypical populations. And last of all, thank you to Open Science because it helped make this research possible. Thank you to all my mentors, collaborators, and colleagues and reviewers for making this study possible and helping me grow. Thank you.
Yeah, thank